Good morning dear students. Once again I am here with the next online session, and students, in our previous class we have discussed about the tissues, what is a tissue, histology, the utility of tissues, as well as we have started with the types of plant tissues. And the first type of plant tissue, that is meristematic tissue, we have discussed. Now we will discuss about the second type of plant tissue, that is permanent tissues. So what are permanent tissues? The permanent tissues, they comes from meristematic tissue and they are mature, means they are non-dividing. We know the meristematic ones, they are dividing, they help in growth. So permanent tissues are the ones which are non-dividing and they don't help in the growth. They are made up of dead cells that have lost the power to divide. One more characteristic is they have a definite shape, size and function, and they may be dead or living, but they are non-dividing. Mostly they are dead. The process by which the cells arise from the meristematic tissue and they take up a permanent shape, size and function, that process is called differentiation. Means these permanent ones are also made up from the meristematic ones, which becomes mature and they stop dividing after maturity. That stage they are called as permanent tissues, and that process is called differentiation. Right. Now again we have the subcategories of this permanent tissue: the simple permanent tissue and the complex permanent tissue. Again, in simple permanent tissue, we will discuss about parenchyma, collenchyma and sclerenchyma. And in the complex permanent tissue, we will discuss about the xylem and phloem. But today, I would like to discuss about the first category only, that is simple permanent tissues, the parenchyma, collenchyma and sclerenchyma. Here on the screen, you can see the specific features of all these three. Parenchyma. In parenchyma, the cells are thin-walled. Only primary wall is present. Whereas in collenchyma, there is a thick primary wall at the corners. And third stage, that is sclerenchyma, it is completely thick-walled. Means, now see the differences. Parenchyma, they are thin-walled. Only primary wall is present. Collenchyma, they have thick walls at corners. Sclerenchyma, completely thick wall. Okay, so this is the difference between these three. Now in parenchyma, the cells are loosely arranged, like in gaseous stage, right? They are loosely arranged. In collenchyma, they are compactly arranged. Okay, and same as in sclerenchyma, the cells are compactly arranged. So in parenchyma only they are loosely arranged, like in gaseous states, the particles of matter are loose. Now the third characteristic is, in parenchyma the cells are living and nucleus is present. Same thing is there in collenchyma also, the cells are living, nucleus is seen. But in sclerenchyma the cells are not living, they are dead and they are devoid of the cellular contents. Okay. Better these points keep in your mind, because these are somewhere they are similar, somewhere they are different. So these characteristics of all these three must be in your mind, so that if I ask you only about parenchyma, you would be able to write the points. The next characteristic of parenchyma is they are spherical, they are polygonal, oval, rectangular or rod shape. Now these are all the various shapes in which parenchyma are found. Whereas collenchyma, again the shapes are variable. We cannot specify them that spherical is there, polygonal is there, oval is there. They are variable. They can take any shape. Sclerenchyma, they are always elongated. Okay. Now parenchyma, they have intercellular spaces. What are intercellular spaces? Means the space in between the cells. Obviously, if the cells are loosely arranged, like same, take the example of matter in gaseous stage. If the matter particles were loosely arranged, means they have the spaces, the intercellular spaces. Same thing parenchyma, the cells are loosely arranged, so there is space in between. Whereas in collenchyma and sclerenchyma, the intercellular space is absent. And why it is absent? Because in the second characteristic we have discussed that the cells in the sclerenchyma and collenchyma are closely, compactly arranged. So there is no space. Now the last characteristic is, in parenchyma the cells are vacuolated, means vacuoles are there in the cells. Whereas in parenchyma and sclerenchyma, vacuoles are absent. Okay students, so these are all the characteristics of parenchyma, parenchyma and sclerenchyma. We'll discuss more in detail during the live class also. Till that time, just go through these and keep on revising the things till the online live class, and we'll discuss again in explain during the live class. Okay students, I hope you have understood about these meristematic tissue types, that apical, intercalary and lateral, and now today about the permanent tissue, that is simple permanent tissue types, parenchyma, parenchyma and sclerenchyma. Okay, see you in the next class with the complex permanent tissues, that is xylem and phloem. Till that time, thank you and have a great day.